Whole genome sequencing, Wikipedia audio. Whole genome sequencing is the process of determining the complete DNA sequence of an organism's genome at a single time. This entails sequencing all of an organism's chromosomal DNA as well as DNA contained in the mitochondria and, for plants, in the chloroplast. Whole genome sequencing has largely been used as a research tool, but is currently being introduced to clinics. In the future of personalized medicine, whole genome sequence data will be an important tool to guide therapeutic intervention. The tool of gene sequencing at SNP level is also used to pinpoint functional variants from association studies and improve the knowledge available to researchers interested in evolutionary biology, and hence may lay the foundation for predicting disease susceptibility and drug response. Whole genome sequencing should not be confused with DNA profiling, which only determines the likelihood that genetic material came from a particular individual or group, and does not contain additional information on genetic relationships, origin, or susceptibility to specific diseases. In addition, whole genome sequencing should not be confused with methods that sequence specific subsets of the genome. Such methods include whole exome sequencing or SNP genotyping. History. As of 2017, there were no complete genomes for any mammals, including humans. Between 4% to 9% of the human genome, mostly satellite DNA, had not been sequenced. The DNA sequencing methods used in the 1970s and 1980s were manual, for example Maxim Gilbert sequencing and Sanger sequencing. The shift to more rapid, automated sequencing methods in the 1990s finally allowed for sequencing of whole genomes. The first organism to have its entire genome sequenced was Haemophilus influenza in 1995. After it, the genomes of other bacteria and some archaea were first sequenced, largely due to their small genome size. H. influenza has a genome of 1,830,140 base pairs of DNA. In contrast, eukaryotes, both unicellular and multicellular such as amoeba dubia and humans respectively, have much larger genomes. Amoeba dubia has a genome of 700 billion nucleotide pairs spread across thousands of chromosomes. Humans contain fewer nucleotide pairs than A. dubia, however their genome size far outweighs the genome size of individual bacteria. The first bacterial and archaeal genomes, including that of H. influenza, were sequenced by shotgun sequencing. In 1996 the first eukaryotic genome was sequenced. S. cerevisiae, a model organism in biology, has a genome of only around 12 million nucleotide pairs, and was the first unicellular eukaryote to have its whole genome sequenced. The first multicellular eukaryote, an animal, to have its whole genome sequenced was the nematode worm, Sinorhabditis elegans in 1998. Eukaryotic genomes are sequenced by several methods including shotgun sequencing of short DNA fragments and sequencing of larger DNA clones from DNA libraries such as bacterial artificial chromosomes and yeast artificial chromosomes. In 1999, the entire DNA sequence of human chromosome 22, the shortest human autosome, was published. By the year 2000, the second animal and second invertebrate genome was sequenced, that of the fruit fly Drosophila melanogaster, a popular choice of model organism in experimental research. The first plant genome, that of the model organism Arabidopsis thaliana, was also fully sequenced by 2000. By 2001, a draft of the entire human genome sequence was published. The genome of the laboratory mouse Mus musculus was completed in 2002. In 2004, the Human Genome Project published an incomplete version of the human genome. Currently, thousands of genomes have been wholly or partially sequenced. Almost any biological sample containing a full copy of the DNA, even a very small amount of DNA or ancient DNA, can provide the genetic material necessary for full genome sequencing. Such samples may include saliva, epithelial cells, bone marrow, hair, seeds, plant leaves, or anything else that has DNA-containing cells. The genome sequence of a single cell selected from a mixed population of cells can be determined using techniques of single-cell genome sequencing. This has important advantages in environmental microbiology in cases where a single cell of a particular microorganism species can be isolated from a mixed population by microscopy on the basis of its morphological or other distinguishing characteristics. In such cases, the normally necessary steps of isolation and growth of the organism in culture may be omitted, thus allowing the sequencing of a much greater spectrum of organism genomes. Experimental details. Single-cell genome sequencing is being tested as a method of pre-implantation genetic diagnosis, wherein a cell from the embryo created by in vitro fertilization is taken and analyzed before embryo transfer into the uterus. After implantation, cell-free fetal DNA can be taken by simple venipuncture from the mother and used for whole genome sequencing of the fetus. Sequencing of nearly an entire human genome was first accomplished in 2000 partly through the use of shotgun sequencing technology. While full genome shotgun sequencing for small genomes was already in use in 1979, broader application benefited from pairwise end sequencing, known colloquially as double-barrel shotgun sequencing. As sequencing projects began to take on longer and more complicated genomes, multiple groups began to realize that useful information could be obtained by sequencing both ends of a fragment of DNA. Although sequencing both ends of the same fragment and keeping track of the paired data was more cumbersome than sequencing a single end of two distinct fragments, the knowledge that the two sequences were oriented in opposite directions and were about the length of a fragment apart from each other was valuable in reconstructing the sequence of the original target fragment. The first published description of the use of paired ends was in 1990 as part of the sequencing of the human HPRT locus, although the use of paired ends was limited to closing gaps after the application of a traditional shotgun sequencing approach. The first theoretical description of a pure pairwise end sequencing strategy, assuming fragments of constant length, was in 1991. In 1995, the innovation of using fragments of varying sizes was introduced, and demonstrated that a pure pairwise end sequencing strategy would be possible on large targets. The strategy was subsequently adopted by the Institute for Genomic Research to sequence the entire genome of the bacterium Haemophilus influenza in 1995, and then by Celera Genomics to sequence the entire fruit fly genome in 2000, and subsequently the entire human genome. Applied Biosystems, now called Life Technologies, manufactured the automated capillary sequencers utilized by both Celera Genomics and the Human Genome Project. While capillary sequencing was the first approach to successfully sequence a nearly full human genome, it is still too expensive and takes too long for commercial purposes. Since 2005, capillary sequencing has been progressively displaced by high-throughput sequencing technologies such as Illumina dye sequencing, pyrosequencing, and SMRT sequencing. All of these technologies continue to employ the basic shotgun strategy, namely, parallelization and template generation via genome fragmentation. Other technologies are emerging, including nanopore technology. Though nanopore sequencing technology is still being refined, its portability and potential capability of generating long reads are of relevance to whole genome sequencing applications. In principle, full genome sequencing can provide the raw nucleotide sequence of an individual organism's DNA. However, further analysis must be performed to provide the biological or medical meaning of this sequence, such as how this knowledge can be used to help prevent disease. Methods for analyzing sequencing data are being developed and refined. Because sequencing generates a lot of data, its output is stored electronically and requires a large amount of computing power and storage capacity. Cells used for sequencing. Early techniques. While analysis of WGS data can be slow, it is possible to speed up this step by using dedicated hardware. Current techniques. Analysis. Commercialization. Incentive. History 2. A number of public and private companies are competing to develop a full genome sequencing platform that is commercially robust for both research and clinical use, including Illumina, Gnome, Sycamore, 454 Life Sciences, Pacific Biosciences, Complete Genomics, Helicos Biosciences, GE Global Research, Affymetrix, IBM, Intelligent Biosystems, Life Technologies, and Oxford Nanopore Technologies. These companies are heavily financed and backed by venture capitalists, hedge funds, and investment banks. A commonly referenced commercial target for sequencing cost is the $1,000 genome. In October 2006, the X Prize Foundation, working in collaboration with the J. Craig Venter Science Foundation, established the Archon X Prize for Genomics, intending to award $10 million to the first team that can build a device and use it to sequence 100 human genomes within 10 days or less, with an accuracy of no more than one error in every 1 million bases sequenced, with sequences accurately covering at least 98% of the genome, and at a recurring cost of no more than $1,000 per genome. Comparison with other technologies. The Archon X Prize for Genomics was cancelled in 2013, before its official start date. In 2007, Applied Biosystems started selling a new type of sequencer called Solid System. The technology allowed users to sequence 60 gigabases per run. In June 2009, Illumina announced that they were launching their own personal full genome sequencing service at a depth of 30 times for $48,000 per genome. In August 2009, the founder of Helicos Biosciences, Stephen Quake, stated that using the company's single-molecule sequencer he sequenced his own full genome for less than $50,000. In November 2009, Complete Genomics published a peer-reviewed paper in Science demonstrating its ability to sequence a complete human genome for $1,700. In May 2011, Illumina lowered its full genome sequencing service to $5,000 per human genome, or $4,000 if ordering 50 or more. Helicos Biosciences, Pacific Biosciences, Complete Genomics, Illumina, Sycamore, Ion Torrent Systems, Halcyon Molecular, Nabsys, IBM, and GE Global appear to all be going head-to-head in the race to commercialize full genome sequencing. With sequencing costs declining, a number of companies began claiming that their equipment would soon achieve the $1,000 genome. These companies included Life Technologies in January 2012, Oxford Nanopore Technologies in February 2012 and Illumina in February 2014. DNA microarrays. However, as of 2015, the NGRI estimates the cost of obtaining a whole genome sequence at around $1,500. Full genome sequencing provides information on a genome that is orders of magnitude larger than by DNA arrays, the previous leader in genotyping technology. Applications. For humans, DNA arrays currently provide genotypic information on up to 1 million genetic variants, while full genome sequencing will provide information on all 6 billion bases in the human genome, or 3,000 times more data. Because of this, full genome sequencing is considered a disruptive innovation to the DNA array markets, as the accuracy of both range from 99.98% to 99.999% and their consumables cost of $5,000 per 6 billion base pairs is competitive with DNA arrays. Whole genome sequencing has established the mutation frequency for whole human genomes. The mutation frequency in the whole genome between generations for humans is about 70 new mutations per generation. An even lower level of variation was found comparing whole genome sequencing in blood cells for a pair of monozygotic 100-year-old centenarians. Only 8 somatic differences were found, though somatic variation occurring in less than 20% of blood cells would be undetected. Mutation frequencies. Genome-wide association studies. Diagnostic use. In the specifically protein-coding regions of the human genome, it is estimated that there are about 0.35 mutations that would change the protein sequence between parent/child generations. In cancer, mutation frequencies are much higher, due to genome instability. This frequency can further depend on patient age, exposure to DNA-damaging agents and the activity/inactivity of DNA repair mechanisms. Furthermore, mutation frequency can vary between cancer types. In germline cells, mutation rates occur at approximately 0.023 mutations per megabase, but this number is much higher in breast cancer, in lung cancer or in melanomas. Since the haploid human genome consists of approximately 3,200 megabases, this translates into about 74 mutations in germline DNA per generation, but 3,776 to 5,312 somatic mutations per haploid genome in breast cancer, 56,640 in lung cancer and 105,600 in melanomas. The distribution of somatic mutations across the human genome is very uneven, such that the gene-rich, early replicating regions receive fewer mutations than gene-poor, late replicating heterochromatin, likely due to differential DNA repair activity. In particular, the histone modification H3K9Me3 is associated with high, and H3K36Me3 with low mutation frequencies. In research, whole genome sequencing can be used in a genome-wide association study, a project aiming to determine the genetic variant or variants associated with a disease or some other phenotype. In 2009, Illumina released its first whole genome sequencers that were approved for clinical as opposed to research-only use, and doctors at academic medical centers began quietly using them to try to diagnose what was wrong with people whom standard approaches had failed to help. The price to sequence a genome at that time was $19,500, which was billed to the patient but usually paid for out of a research grant. One person at that time had applied for reimbursement from their insurance company. For example, one child had needed around 100 surgeries by the time he was 3 years old, and his doctor turned to whole genome sequencing to determine the problem. It took a team of around 30 people that included 12 bioinformatics experts, 3 sequencing technicians, 5 physicians, 2 genetic counselors and 2 ethicists to identify a rare mutation in the XIAP that was causing widespread problems. Currently available newborn screening for childhood diseases allows detection of rare disorders that can be prevented or better treated by early detection and intervention. Specific genetic tests are also available to determine an etiology when a child's symptoms appear to have a genetic basis. Full genome sequencing, in addition, has the potential to reveal a large amount of information that is currently not completely understood, may not be clinically useful to the child during childhood, and may not necessarily be wanted by the individual upon reaching adulthood. Due to recent cost reductions, whole genome sequencing has become a realistic application in DNA diagnostics. In 2013, the 3GB Test Consortium obtained funding from the European Union to prepare the healthcare system for these innovations in DNA diagnostics. Quality assessment schemes, health technology assessment and guidelines have to be in place. The 3GB Test Consortium has identified the analysis and interpretation of sequence data as the most complicated step in the diagnostic process. At the consortium meeting in Athens in September 2014, the consortium coined the word genotranslation for this crucial step. This step leads to a so-called geno-report. Guidelines are needed to determine the required content of these reports. Genomes to People, an initiative of Brigham and Women's Hospital and Harvard Medical School, was created in 2011 to examine the integration of genomic sequencing into clinical care of adults and children. G2P's director, Robert C. Green, had previously led the REVEAL study Risk Evaluation and Education for Alzheimer's Disease, a series of clinical trials exploring patient reactions to the knowledge of their genetic risk for Alzheimer's. The introduction of whole genome sequencing may have ethical implications. On one hand, genetic testing can potentially diagnose preventable diseases, both in the individual undergoing genetic testing and in their relatives. On the other hand, genetic testing has potential downsides such as genetic discrimination, loss of anonymity, and psychological impacts such as discovery of non-paternity. Some ethicists insist that the privacy of individuals undergoing genetic testing must be protected. Indeed, privacy issues can be of particular concern when minors undergo genetic testing. Illumina's CEO, Jay Flatley, claimed in February 2009 that by 2019 it will have become routine to map infants' genes when they are born. This potential use of genome sequencing is highly controversial, as it runs counter to established ethical norms for predictive genetic testing of asymptomatic minors that have been well established in the fields of medical genetics and genetic counseling. The traditional guidelines for genetic testing have been developed over the course of several decades since it first became possible to test for genetic markers associated with disease, prior to the advent of cost-effective, comprehensive genetic screening. When an individual undergoes whole genome sequencing, they reveal information about not only their own DNA sequences, but also about probable DNA sequences of their close genetic relatives. This information can further reveal useful predictive information about relatives' present and future health risks. Hence, there are important questions about what obligations, if any, are owed to the family members of the individuals who are undergoing genetic testing. In Western/European society, tested individuals are usually encouraged to share important information on any genetic diagnoses with their close relatives, since the importance of the genetic diagnosis for offspring and other close relatives is usually one of the reasons for seeking a genetic testing in the first place. Nevertheless, a major ethical dilemma can develop when the patients refuse to share information on a diagnosis that is made for serious genetic disorder that is highly preventable and where there is a high risk to relatives carrying the same disease mutation. Under such circumstances, the clinician may suspect that the relatives would rather know of the diagnosis, and hence the clinician can face a conflict of interest with respect to patient-doctor confidentiality. Privacy concerns can also arise when whole genome sequencing is used in scientific research studies. Researchers often need to put information on patients' genotypes and phenotypes into public scientific databases, such as locus-specific databases. Although only anonymous patient data are submitted to locus-specific databases, patients might still be identifiable by their relatives in the case of finding a rare disease or a rare missense mutation. The first nearly complete human genomes sequenced were two Americans of predominantly Northwestern European ancestry in 2007 and James Watson at 7.4-fold. This was followed in 2008 by sequencing of an anonymous Han Chinese man, a Yoruban man from Nigeria, and a female Caucasian leukemia patient. Steve Jobs was among the first 20 people to have their whole genome sequenced, reportedly for the cost of $100,000. As of June 2012, there were 69 nearly complete human genomes publicly available. In November 2013, a Spanish family made their personal genomics data publicly available under a Creative Commons public domain license. The work was led by Manuel Corpaz, and the data obtained by direct-to-consumer genetic testing with 23andMe and the Beijing Genomics Institute. This is believed to be the first such public genomics dataset for a whole family. Ethical concerns. People with public genome sequences. Sequenced genomes.